Hi, my name is Braydon Lu, and I'm from a high school in California. Our research topic was about identifying conserved nucleotides of microRNA targeted seed sequences for their regulated genes in TCGA cancers. MicroRNAs are small RNAs that help regulate gene expression. A particular sequence in the microRNA, called the seed sequence, can bind to a seed sequence in the 3' UTR of mRNA, which can prevent or reduce translation by speeding up the degradation of that mRNA. SNPs, also known as single nucleotide polymorphisms, are a type of mutation where a single nucleotide may change. This can cause the messenger RNAs to not be able to bind to the microRNAs properly, which can cause dysregulation. This gene dysregulation can lead to many different diseases, including cancers. Our research question was whether or not we can find any conserved nucleotides in the seed sequences of microRNA gene pairs with inverse correlation between tumor and normal samples. From a previous study, we were able to retrieve 238 such pairs, which were already categorized into 8 different TCGA cancers. We used the same microRNA target prediction databases used to identify the pairs from the previous study to find their predicted target seed sequences. After we found our predicted seed sequences, we had to look through each pair to check if the seed sequences for that pair were in consensus. How we did this was, in order for there to be a consensus sequence for that pair, either only one tool generated a sequence for us, or 2 or more tools supported the same seed sequence. We chose to disregard any pairs that did not have a clear consensus seed sequence. After finding our consensus seed sequences, we ran all of them through WebLogo, which gave us logos for each type of cancer for the microRNA and mRNA seed sequences. Using these logos, we were able to identify areas where a nucleotide would be especially prevalent, which could indicate that it is conserved. Before generating the logos for each cancer, we split the microRNA gene pairs by their tumor correlation coefficient. What this basically indicates is the relationship between the microRNA and the gene in tumor samples, which in turn also indicates the relationship between the microRNA and gene in normal samples, since it is the inverse of the relationship in tumor samples. The left side are the logos we generated using pairs with a negative tumor correlation coefficient. And on the right side, we generated logos with the pairs with the positive tumor correlation coefficient. In summary, conserved nucleotides of their gene microRNA seed sequences exist mostly in pairs with the positive tumor correlation coefficient. This is the case for most cancers except for HNSC, where the conserved nucleotides are actually found in the pairs with a negative tumor correlation coefficient. Overall, what our results indicate to us is that microRNAs have likely changed their original regulatory roles on their targeting genes in cancer patients. Our next research will be evaluating the functional consequences of these conserved nucleotides. Here are some of the resources we used. Thank you for listening to my presentation. If you have any questions, I would be happy to answer them in the chat.